Okay, so last but not least, we have Morena, or Marina, sorry if I'm saying this wrong,
Morente.
um, who is currently a postdoc at the University of Oxford, and she will be presenting her previous work done at the University of Granada in Spain, which was recently published in Annals of Applied Biology. So take it away.
Thank you. It's a pleasure to be here. I'm going to share the presentation. Okay. Okay, perfect. Can you see the presentation?
Yeah, perfect.
Okay. So yeah, I'm coming from the University of Oxford, but this work about the effect of semi-tilling on the arthropod community in olive groves was developed at the University of Granada. And first of all, yeah, I'm going to move. Oh, it's not moving. No, it's frozen. I'm sorry. It's frozen.
If you click on it,
Okay.
and then move it, it might work. If you click elsewhere, it sometimes changes what you're going to move.
Okay, yeah, that is. Thank you.
Right.
So, yeah, first of all, to say that the ground cover management in olive groves historically has relied on the removal of weeds to prevent the water and nutrient competition with the tree. This removal has been made by tillage or the use of herbicides. And with the advent of mechanization, the impact of these practices increased. And as you can see here, the erosion and the runoff had a big impact, giving results like this dramatic picture, as you can see in the picture. So, to decrease this impact of tilling, different alternative methods are applied nowadays, and two of the more common methods are semi-tilling and mowing. In the first picture, you can see an olive grove under semi-tilling, and in the second one grove under mowing. In these two practices, the common thing is that they left to grow the vegetation during winter, but the removal of the vegetation in spring is different. In the semi-tilling, they remove totally one part of the vegetation. It depends on the farmer, but as you can see here in the first picture, they remove the vegetation behind the trees, leaving the vegetation in the streets. But in other cases, they remove the vegetation in the streets, leaving the vegetation behind the olive tree canopy. And they use tillage and herbicides as well. And this is a method very common used in conventional and integrated olive groves. And on the other hand, in mowing, this practice consists of the cutting the entire vegetation in the ground cover, leaving a small surface, a thin surface of vegetation, and the plant debris are incorporated to the ground cover as well. And this kind of practice is common in organic olive groves. Well, the removal of vegetation can affect environmentally not only the soil structure, but other factors such as biodiversity and ecosystem services that biodiversity provides, such as pest control. In olive groves, this kind of impact on biodiversity has been widely studied, but always comparing between mowing olive groves and totally tilled olive groves. But this kind of intermediate practice, such as the semi-tilling, has been overlooked. So, in our study, we were interested in the study of the arthropods in the different strata of the crops, the tree canopy, the ground cover, and the soil, because arthropods can interchange between the different strata, forming complex networks of interactions between them. Then we decided to move one step forward, and to study the effect of semi-tilling under an ecological network perspective. And The objectives of the study were to assess the impact of the ground cover management, in our case semi-tilling and mowing, on the richness and abundance of arthropods, the trophic interactions between them and the basal resources used, the arthropod food web structure, that is a measure of the stability of the system, and the arthropod distribution among the crop strata. And we developed this study in the province of Granada, southern Spain, as you can see in the map, where we used two different olive groves under the same environmental conditions. These crops were managed by a public agency, so we had to adapt our design of sampling to the conditions of the agency. And we had a mowed olive grove and a semi-tilled olive grove. But the better option would be to have the two treatments in the two plots, but it wasn't possible. So we sampled the 3 strata of the crops. We used the branch beating for the olive canopy, sweep net for the ground cover, and pitfall traps for the soil arthropods. We identified every arthropod found in our sampling and constructed the different networks. These networks were constructed by crop strata and month-on sampling, because we were sampling between May and June. And we used 2 different approaches: the information found in literature and the analysis of stable isotopes. And we included the most abundant pests in these crops in concrete, that were the moth, the olive moth, that is Preisoleae, and the olive scilla, that is Eophilura olivina. And I have said before, we used the stable isotope analysis to identify trophic interactions and to construct the network. This technique is based on the differences in assimilation between the heavy isotopes carbon-13 and nitrogen-15 in relation to the lighter carbon-12 and nitrogen-14. So when a predator prey on a basal resource, these heavy isotopes are stored in its tissue, and the lighter isotopes are excreted. With this information, we can identify basal resources in the trophic network and the trophic position of the different taxa in the community. And moreover, we calculated different classical structure descriptors that were related with the number of nodes in the network and the number of links per node and their identity in the trophic web. And moving to results, here you can see an example of the networks that we obtained. They were 2 networks from the ground cover, the first one from the semi-tilled groove and the other one from the mowed groove. And we detected changes in the arthropod composition. That means that we had similar number of nodes, as you can see here. We have 3 different trophic levels. These are parasitoids in yellow, predators in orange, and herbivores in green. The number of nodes were similar, but the identity of some of them was different. That is a very interesting result, because the ecological function of these nodes can be different between the 2 treatments. But on the other hand, we didn't find any effect on species richness or abundance, and we didn't find any effect on network structure. This last result is due of the predators in olive groves. Most of them are generalist, so they can use different basal resources regardless the prey availability. So they can maintain the number of links that they have in the network and maintain the network integrity and structure in these terms of number of links. Moreover, and interestingly, we identified or we observed a decrease in the pest abundance, Euphylura olivina and Priceoreae, in mowed groves. So what's happening here? Using the carbon and nitrogen isotope signature, we identified changes in the arthropod diet in 3 of the most important predators in olive groves in terms of pest control, that were Chrysoperla carnea, Anthocoris nemoralis, and Lycosa fascidiventris. As you can see here, the carbon-13 is very different in the different strata between the 2 treatments, which indicate this change in basal resources. Sadly, these are very generalist predators, and we couldn't identify the specific changes that they did because it is necessary molecular analysis to do that, but it would be interesting to do that in the future. But in the case of Anthocoris nemoralis, using this isotopic signature, we saw or we detected trophic interactions with one of the pests, Euphylura olivina. And in the mowed groove, we saw that both Anthocoris nemoralis from the canopy and the ground cover, they were feeding on Euphylura olivina. Meanwhile, in the semi-till, just Anthocoris nemoralis from the canopy were feeding on the pests. What happened with Anthocoris nemoralis in the ground cover? We found Anthocoris nemoralis in the ground cover as well, but the isotopic distances with Euphylura olivina were too far. So we have here 2 scenarios. Maybe they are eating other resources from the vegetation cover, and they are not moving to the canopy because they don't have this corridor of vegetation to move to the canopy, or maybe they are eating some Euphylura olivina but in a low rate. And these 2 scenarios means that we have a greater predation pressure in mowed olive groves. Finally, we wanted to assess as well the effect of the ground cover management on the distribution of the arthropods in the strata of the olive groves, the canopy, the ground cover, and the soil. And we saw that the vegetation, the ground cover management didn't have any effect on the distribution. Just the crop strata has this kind of effects on the distribution of the different species in the crop. We wanted to see if we had a seasonal tendency or seasonal changes, but the results were the same. We found that the crop stratum has the effect on the different distribution of the species in the crop. So to conclude, we can say that semi-tilling has an effect on species composition in the arthropod community, which can be affecting to the ecological functions of this arthropod in the different managements. Moreover, we observed changes in basal resources that affect predators' diets, and these results can be related to the effect on pest abundance. So we observed a decreased pest control in semi-tilled olive groves. So mowing, we can conclude that mowing enhance the pest control of the main pests in olive groves. But we have to say that we didn't find any effect on community structure or distribution of the arthropods. So this practice, semi-tilling, is a better practice than the entire plugging of the ground cover, and it can be used as a transitional method between the conventional methods of entirely plugging to the mowing one. And that's everything. I would like to thank to my co-author, Francisca Ruano, and the technician, Francisco Sigeo, and thank you for your attention.
Thank you, Marina. That's really fantastic stuff.
Thank you.
Leave a few minutes for some questions to come in. Maybe I'll start us off. Does this do you know if this work kind of translates from olive groves to other orchard crops?
Mhm. Yeah, I don't know because yeah, at the olive grove, maybe to other tree crops like stone fruits or these kind of things, it could be translated. I think it's the same system because you have these three strata where insects are moving. But in the case of orchards, yeah, it would be interesting to make the comparison at some point because I think the structure of the system is different.
Yeah. Okay, brilliant. Oh, we do have a hand raised. Javier, please, I will ask you to unmute.
Yeah. Yeah. Hello.
Hi.
Can you hear me? Hello. Another University of Granada student. It's nice to
Ah,
see you.
nice
Uh,
to see you.
yeah. Back to everybody's here in the UK at the end. So just
Yes.
a quick question. Did you see any effect in the olive oil later on with this two
Oh,
different?
yeah, no, I don't know. We didn't analyze that. Yeah.
Right.
Yeah, that
Makes
would be
sense.
interesting, but yeah, I can tell you maybe in other similar samplings that we have done, we know that we have differences in the yield, in
Right.
the olive yield that can be related with the olive
Mm.
production. Yeah, at the
Right.
end. Hmm.
Makes sense. Thank you.
And so, can I ask a question, Jordan, please?
Yes, please.
Yeah, so Marina, I wanted to ask, so I mean, you showed that there wasn't much change in the the web of arthropod interactions, but can you say anything about the potential changes in olive pests, olive tree pests, arthropod pests amongst your results there?
Yeah, the thing is that yeah, we detected these changes in the abundance of olive pests.
Okay.
Yeah, the structure of the network wasn't affected because predators can take very easily of resources. So yeah, if we are focused on the number of interactions that we have in the food web, we didn't detect these changes. But the most interesting thing is that we detected these changes in the identity of the node that can be affecting the interactions that the pest has with the different predators or parasitoids, and as well these changes in the basal resources of some of the predators. The thing is that we would like to move one step forward and to make molecular analysis because of the gut content, because it would be interesting to specify which are the changes that arthropods or predators in this case are doing between the two treatments. But definitely there is an effect in the abundance of the pests because the two of them has like was halved in the mode groups. That is a very huge differences between them.
Okay, great. Thank you.
You're welcome.
Great. Well, I think that concludes today's webinar. I'd like to join um, everyone in saying well done to all our speakers. A big round of applause. Um, yeah, and as I say, Marina's paper is now on Annals of Applied Biology, so you can read it if you're interested to find out more. If you have any questions for any of our speakers, please do send them an email. And thank you on behalf of the AAB for joining us today for this webinar series, and we hope to see you next month.